So my name is Guy Polturak. I'm a postdoc at Professor Anne Osborne's group at the John Innes Center in the UK. And I'm going to be talking about genomics-driven discovery of defense-related gene clusters in wheat. But I think as a representative of the plant sciences today, I should probably start with a bit of a background about plants. Sorry, so one thing we all know about plants is that plants can't move. So if you're a plant and you're under threat from a herbivore such as this, you can't really run away. So plants have to fight back. And one of the main ways for them to do this is with chemistry, which is why plants are considered to be nature's best chemists and produce hundreds of thousands of different molecules. And these molecules allow the plants to react to and interact with their environment. These molecules are made by enzyme-coding genes, which together form biosynthetic pathways in which a precursor molecule is converted to an end product in a sequence of reactions. And in recent years, we find more and more examples of biosynthetic pathways in which genes are actually found together on the genome, forming what is called biosynthetic gene clusters, something which resembles bacterial operons, for example. And this phenomenon, which is interesting in itself in terms of the evolutionary aspect, it's also a very powerful tool to discover new plant chemistry and elucidate pathways. So for example, traditionally, plants which have interesting activities would be studied. The metabolites that are responsible for these activities would be identified by chemical analysis tools. And finally, the genes which make these metabolites would be characterized. But this phenomenon of gene clustering allows us to take a sort of reverse approach in which we first look at the genome of a plant of interest to look for interesting looking gene clusters in the hope that these gene clusters will eventually lead us to interesting metabolites. So this approach, which is sometimes called genome mining, is quite prevalent in microbiology, for example, for searching for novel antibiotics, but only now is emerging as a valid approach to elucidate new plant chemistry. So to utilize this approach, the first thing you need is a sequence genome for your plant. So in the case of wheat, which is obviously super reported in agriculture, the genome was actually only published 2 years ago. And so we leverage this discovery of the genome to make new discoveries in the secondary metabolism of wheat, which is surprisingly not so well characterized. So in our project, we used the previously published co-expression network analysis, which is based on a collection of multiple RNA-seq data sets from wheat. And this analysis assigned all of the wheat genes into 6 subsets, and each subset into multiple modules based on the gene expression. So we hypothesized that by mining one of these subsets, which is called disease, we can find new genes and new molecules which are related to plant defense. So this disease subset basically includes all of the wheat genes which have any sort of expression pattern in experiments in which wheat was challenged by different pathogens or pathogen effectors. So to do this, we calculated the average gene expression delta between each experiment and its control for all the different pathogens in the data set per each module. And we then sorted these modules by the average delta across all experiments. So basically what you have is that the top ranking modules are the ones which include genes which are mostly induced, the most highly induced by pathogens. And if you look closer at these modules, you can see that the induction is consistent across all different experiments, which shows that these genes are not involved in a response to a specific pathogen, but more of a general defense response. So we next wanted to see if we can really mine this set of genes to find new putative gene clusters. And we did actually find quite a few. which potentially produce triterpenes, diterpenes, and isoflavonoids, which are classes of compounds which are all have been implicated in plant defense. So we then focused on one of these clusters, which was a putative triterpene cluster. We cloned all of the genes in the cluster, and we used the transient expression system, which is based on infiltration of agrobacteria into a tobacco-related plant. And indeed, we could see that these genes form a biosynthetic pathway, which eventually results in production of a triterpene, which is based on an isoborinol scaffold. So this system allowed us also to produce enough of this compound to be able to purify it and elucidate its structure by NMR analysis. So we did that, and we found that this compound is actually a novel compound which was not previously reported in wheat or in other plants. So we then looked further into the cluster, and looking at the expression of the cluster genes, we found that the genes are only expressed after exposure to pathogens, as you can see marked in red here. And correspondingly, we could only detect Ellarinacin in wheat leaves after treatment with the elicitor methyl jasmonate. So this sort of strengthens the notion that this compound has a defense-related role, but also is a nice example of why this sort of compound would not be able to, we would not be able to identify by just simple chemical screening of wheat extracts. So if Ellarinacin does have a role in plant defense, then you would expect that plants that lack this compound would be more susceptible to disease. So we used a model in which we infect wheat leaves with a powdery mildew, which is a fungal pathogen. And we can see that the mutants for this pathway are more susceptible to the disease compared to their controls. So now that we found this defense-related cluster in wheat, we wanted to know if this is something which is specific to wheat or can be found in other plant species. So the first place to look is obviously the most related species. So wheat has two ancestors which also have a sequenced genome. One of them is the wild emmer wheat, which is the donor of the A and B genomes, and one of them is Aegilops, which is the donor of the D genomes. And indeed, we could see that the cluster is found in these two plants, and also that it's quite highly conserved. And we also found a homologous-looking cluster in another grass called Brachypodium distachyon. And also in oat, we could see that two of the genes from the cluster are paired together, forming what looks like a sort of early version of this cluster. Now, if we look at other plants from the Poaceae family, for example, sorghum, maize, rice, millet, the cluster is not found on these genomes. And if you look at the phylogeny of this family, this kind of makes sense because you can see that the plants that have this cluster, oat, wheat, and Brachypodium, are all part of this subfamily, which is called Poideae. And the more distant species do not have this cluster. So it looks like this cluster is probably limited or restricted to this specific subfamily. We then looked further into the homologous-looking cluster from Brachypodium distachyon to see, first of all, if it really makes an active cluster, and also if it shares any commonalities with the wheat cluster, which I presented. So we used the same methodologies of transient expression, and we could see that in this case too, these genes do form a sequential biosynthetic pathway, which also begins with the same triterpene scaffold called isoborinol, but the chemistry basically diverges in this pathway, and you end up with a different product, which we also purified and analyzed the structure by NMR. And we also find that in this case, it's a novel structure, which we named Barkinazin. Also, similarly to wheat, when you look at the genes in this cluster, you can see that they're also induced by fungal pathogens, in this case, Fusarium, for example, and also that the levels of Barkinazin and isoborinol are also highly increased following treatment with methyl jasmonate. So this shows that similarly to wheat, the cluster in Brachypodium also has a defense-related role. So to summarize, we saw that genes for arborinant-type triterpene biosynthesis are clustered together both in wheat and the Brachypodium genomes. We see that these clusters play a likely role in chemical defense from pathogens. And this demonstrates that this sort of genomics-driven approach is useful for unraveling difficult to access chemistry or elusive chemistry in plants. And obviously this can be implemented not only in wheat, but in many other plants. For example, for discovery of more defense-related genes and compounds in other crops, or looking into bioactive compounds and their pathways in medicinal plants, and so on. So I'd just like to finish by acknowledging all the people who contributed to this project. So my colleagues at the Ann Osborne Group at the John Innes Center, all of our collaborators at the John Innes Center and the Sainsbury Laboratory, the funders of the fellowships that supported this project. And I also like to thank the organizers of this event, and anyone who's been listening, thank you for listening.
Thank you, Guy. Great talk. Do we have any questions?
So, Guy, beautiful talk. So first, where is this, in which stage of publication is this?
I would say
it's not because I'm so eager for publication. I think you should say something about this. Okay.
Yeah, so it's basically manuscript in preparation, which is actually why I couldn't reveal some of the details. I don't know if some of you noticed, like the structures and so on.
Yes.
Yeah, so we're preparing now.
Now the scientific question. Can this be used also in humans?
Sorry, can this be used on humans?
No, this strategy that you're looking at clusters, clusters in humans, you also have clusters and you need defense from bacteria and pathogens. Is there any relevance?
Not so much, because it seems like this phenomenon of clustering is really not prevalent in mammals. So it's very, very prevalent in bacteria in the form of operons, of course. It's very common in fungi. In plants, there are quite a few examples, but as far as I know, in mammals and other animals, not really.
Thank you. Yeah. Good luck.
I have a question, if I may, a short one. From someone who doesn't know anything about plants. Is this thing can this thing be engineered into other plants to have them, be protected against fungi?
Yeah, absolutely. So that's one of the sort of long-term goals when you find an interesting compound with bioactivity, is to be able to introduce it to another plant. So technology-wise, this is very possible, but in terms of making finally a commercial product from there, it has a lot of problems which has to do with, genetic engineering in plants, and the sort of public opinion about this issue, the very strict regulation and high costs and so on. So that will be the main issue.